everyone, welcome back to your PW English Medium 9th and 10th YouTube channel. I am Asa Suryavanshi, your biology tutor, your biology educator, and today we are back with one shot of this amazing chapter, which is heredity and evolution. Yes, this whole class will be purely in English since it is your English medium batch. So yes, at single go, you're going to find this chapter completely in front of you. I hope you are set with your copies and pens so that you can make your notes right here, right now, along with me. So before I start with this amazing chapter, which is gonna score you easily 4 to 5 marks in your board exams, isn't it great? I feel this is amazing. So if I talk about it, so basically from this chapter, heredity and evolution, the evolution part is excluded. The evolution part is not coming. The evolution part is deleted, right? So remember, we need to cover only and only the part that is heredity, right? Remember this point. Do not get confused. The heredity part will be covered. We will be discussing in today's class, will be part of your board exams. But if you talk about the evolution, this part is deleted. So my dear Spartans, are you guys ready to start with the heredity part of this chapter? If you are, let's get started. So the very first thing we have to see is what we're gonna cover today, right? What is the goal for today's class? So today we're going to look into some of the genetical terms, then we'll be moving towards a monohybrid, dihybrid, and finally we'll conclude the chapter with the last topic, that is sex determination. Yes, towards the end we're going to discuss few questions which have come in the previous year boards, so that you have the idea what sort of questions come from this chapter and how to write answers of this chapter, right? So let's get started without wasting much of our time. Let's get started. The very first thing is this term variation. Now you see the word heredity, but to understand various terms like heredity, genetics, we must be knowing this term variation. Now I hope you all have studied this chapter, how do organisms reproduce, because if you haven't, you need to go and revise it, because this chapter will be based on that chapter, right? We'll be taking many clues from that chapter. So if you have not studied that chapter yet, we have already recorded the video for it, so you can have a look from it, right? So yes, variation, the very first thing, we have discussed this in how do organisms reproduce as well. So variation is nothing but the changes, the differences, right? You see your sibling and you see your brother, you see your sister. Do you see, yes, there is some similarity among you? Yes, we look similar, but do you guys look identical? Obviously you do not. So there is some differences, there is some changes, and those differences are called as variation. Even you look into the individuals of the same population, right? If you are having a look at different humans, yes, you might look similar, but there will be some differences, and that difference is nothing but variation. So variations are nothing but the differences which can be seen in the characters or the traits among the individual of the same species and the offspring of the same parent. Example, the free and the attached earlobe. So we have here, so if you see here, though I am wearing these earrings, but if you see here, the earlobe is quite hanging, right? Could you see it's a hanging earlobe, right? So this is the earlobe. What is earlobe? Basically the lowermost part of your ear, right? The lowermost part of the ear is called as the earlobe. This is your earlobe. And if you look into the earlobe, this one is hanging. This one is hanging. So we call such kind of earlobe as the free or hanging earlobe. But if it is attached to the head, then we call such type of earlobe as attached earlobe. Attached earlobe. It is attached to the head. That is why two sorts of varieties is there. Two sorts of changes are seen in the case of population of humans in the earlobe, right? So this is nothing but variation. Now in the previous chapter we discussed that how these variations will be majorly seen in the case of the sexual reproduction as compared to the asexual reproduction. So in case of the organism which are reproducing sexually, we can see the variation is more. So in sexually reproducing organism, they show greater variation, right? Now why they show greater variation, I told you in that chapter also. This is because inaccuracy in the DNA copying. Also the chromosomes, they are inherited from the two parents, that is the male and the female parent. And also we saw one more point, that there is recombination, right? The chromosome, they get recombination during the process of meiosis. So because of these three steps, the variation is there, right? And I told you these variation generally they are advantageous, they are benefit for the organisms. How they are benefit for the organisms? Let me tell you how, right? So for example, what I want to tell you that these variations, they can either be advantageous, they can be good for the individual, or they can be disadvantageous. Not always they are good for the individuals, they can be bad as well. So there can be disadvantage of the variations also. Variation is nothing but the changes we have seen, like the changes in the earlobe, right? Let us look how they can be advantageous. Let us say like this is the water, and in this water, let's say there is a X named population living of bacteria. So this is a population of bacteria which is living in this water, right? And this water is like around maybe 25 degrees Celsius, let's say, right? But now let us say the temperature increases. The water temperature increases, let us say because of global warming, right? Because of some global warming, this temperature is increased. Now just imagine if temperature is increased, there can be two situations. Either the organisms are going to die. They are going to die. Right? Why? Because this group of bacteria, they are sensitive to this high temperature. They cannot survive in this high temperature. Now, can few of these bacteria survive in this high temperature? Yes, they can, only if they have some changes in them, only if they have some variations in them, the variation which help them to adapt in this changing environment, the variation which led them to survive even in this high temperature, that is, they are becoming resistant to heat. So if yes, such variation is there which allow them to be resistant to heat, they can definitely survive. So I can say yes, some organisms, few of the organisms, they are going to survive. And why they are surviving? Because of variation, because of some changes, right? They are becoming resistant to heat. They are able to survive in this high temperature, right? Obviously these organisms which are dying, they are not showing any variation. I can mention here no variation. So yes, if they are able to have this change which is making them adapting to the changing environment, then it can be good. But can it be disadvantageous? Yes, it can be disadvantageous. Let us say the change is such that, so we all know that this is a cell, and within the cell we have the nucleus where our DNA is present. Within the cell we have a nucleus in which DNA is present. Let us say this change, it has happened in the DNA in such a way that DNA damage has there, or maybe we can say such a drastic DNA change. The DNA is changed. Yes, the change is there, but this change is so drastic that it cannot work with the rest of the cellular apparatus. It cannot work with the rest of the cell. Then obviously such individual will die. So variations can be good, but if variation is so drastic that the DNA cannot function well with the cellular apparatus, then we can say yes, the organism will die. So I can say yes in this case if the DNA changes are drastic, that it is not functioning well, not work with the cellular apparatus, right? The rest of the cytoplasm, it is unable to recognize with it. Then obviously the survival will not be there. The individual will die. Is it clear? So yes, the variations can be good if they help the individual to survive, to adapt, but they can be bad if the DNA change is so drastic that it cannot work well with the cellular apparatus. In that case, that individual won't be able to survive, it's going to die. So we can say yes, these variations, they help the organism to survive. So I can say which sort of reproduction will be better. In the case of variation, I can say yes, sexual mode of reproduction will be better because there will be more variation. We have already discussed that in case of asexual reproduction, the organism which are produced, they are almost similar to the parent, very less variation, right? So I can say here the variation is very less. But here the variation is very much more, which lead to the increase of the survival of the species, right? It will increase the survival of that particular individual. Now let us see what is happening in this particular diagram which is in front of you. So are you able to see, an individual here, right? And this, let us say, is a bacteria which is reproducing asexually. Once it is reproducing asexually, obviously the common body design will be transmitted to the further generation, right? To this generation also the common body design is transferred. But do you see that there are some changes which are incorporated? So can I say these changes, they are incorporated during the reproduction? These variations are there during the reproduction. So yes, there is the passage of the common body design, but there is some change as well. But if we look into the second generation, this was the first generation. If you want, we can write it here. Let us say this is the first generation. And this is the second generation. So could you see that? Yeah. So could you see that there are some changes which are there? Now if we see in the second generation, yes, these same changes, they are inherited from the first generation, but also we could see some altogether new changes. Yes, some older changes were inherited from the previous generation, but there is some new changes. Right? Are you able to see that? So can I say these variation, they are transferred from one generation to another during reproduction, and we can also say these variations, they accumulate generation after generation, right? Suppose this is going to reproduce further, more changes will be there. Suppose this is going to reproduce further, more changes will be there, and there will be a time when there will be production of a species which is not identical to this parent generation at all. That is, there is production of altogether a new species, but obviously it's going to take thousands of years, and this process is called as evolution. Simply, evolution is nothing, I can say variation leads to evolution. These changes, once they accumulate generation after generation, it leads to production of an altogether new species, and this process is evolution. So that was about variation. So yes, they are passed from one generation to another during reproduction, and these variation, they accumulate over the long period of time, which lead to the development of the new species, right? It helps the organism to adapt to different environmental conditions. So yes, basically it helps in the survival of the species in the changing environment. We have discussed this very well here. Let's move ahead. After the topic of variation, we have this term heredity. Now heredity is what? Heredity is nothing but the transmission of character from one generation to another, or simply I can say transmission of character from parents to the offspring, right? So it is nothing but the transmission of the character from the parents to the offspring of the next, right? Also we see this word genetics. This was about heredity, right? This is the heredity. And if I see this word genetics, what is genetics? Genetics is nothing, it's a branch of science which deals with the study of heredity as well as variation. So you know what is heredity? Transmission of character from one generation to another. You know variation, the changes which are there. Now if you are studying it, if you are studying the heredity and you are studying the variation, this process is called as genetics. So genetics is the branch of biology where we are dealing with the study of heredity and variation, right? Let us have a look at the question which has been put here. Why do the children resemble their parents? Have you ever wondered that your hair or maybe eye color looks like your father or let's say your mother? Why is it so? Because we discussed in the last chapter also, because our parents, right, our father and mother give us the chromosomes. Now what does this happen? How does this happen? I'll take this question. First, let me tell you the very basic idea of the last chapter in case you have forgotten. Let me give you a recap of it. So we know that, let's say this is the testes of the father, and this is the ovary of the female mother, right? So testes in the case of the male and ovaries in the case of female. Circle with the plus is the symbol for the female. Circle with the arrow is symbol for male. I have told you in the last chapter, right? So in the case of testes, there will be some cell which will be dividing via the process of meiosis. Yes, you guessed it right. So once they divide via the meiosis, it gives rise to the gametes, that is the sperm cell and the egg cell. Yes. I think now you are able to relate. It gives rise to the sperm and the egg. Obviously, if rest of this cell have 46 chromosomes in them, then the gametes only have, yes, great students, it will only have half the number of chromosomes. That is, it will only have 23 chromosomes. That is, chromosome number is half in the case of the gametes. And once these gametes fuses, once these gametes undergo fertilization, it results in the production of a zygote, which is again having 46 chromosomes. And this will further give rise to a new individual. It will give rise to a new individual. So we could see that we get 23 chromosomes from our mother and 23 chromosomes from our father. So can I say our mother and father contribute equal amount of the DNA, or shall I say the genetic material, to the child? Right, they are contributing equal amount, right? So I can mention here the parent, both the parents, that is the father and the mother, they contribute how much amount? They contribute equal amount, right? It's not like the mother is giving us more or the father is giving us more. So they contribute equal amount of genetic material, that is the DNA, to the offspring, right? To the offspring, to the child. Isn't it? Don't you relate with this particular fact? Don't you relate with this particular fact? Also, once I say we humans have 46 chromosomes, 46 chromosome means how many pair of chromosomes? 46 chromosome means we have 23 pairs of chromosomes. Just like imagine you have 23 pairs of shoes. So if I say you have 23 pair of shoes, for every shoe you have a pair, and that shoe will be like almost identical to it. Right, wherever the design will be on this shoe, it will be on this shoe. Wherever laces will be there, the same lace pattern will be there on the other shoe. But yes, there are other shoes also which might be like your boots, or maybe heels, or maybe flats, or different types of shoes. But if we talk about a pair, they will be almost identical. Similarly, if I say in case of humans, we have, suppose this is a human cell, right? We have, okay, let's erase it. Maybe we have 46 chromosomes. So this is, let's say, first chromosome and its pair. This is the first chromosome and its pair. This is the second chromosome and its pair. And similarly, we have how many chromosomes? We have 23 pair of chromosomes. We have 23 pair of chromosome. That is how many chromosome we are having? We are, ma'am, having 46 chromosome. That is 23 pair. Perfect. 46 chromosome. As in, we have how many pairs? 23 pair. 23 pairs. Do you all agree with me from here? Yes or no? Yes or no? Just tell me. Are you able to relate? Yes, ma'am, or no, ma'am? Yes. So let's see further. Now, if I look into these 23 and if I look into these 23, so there are two sets of chromosome. Again, related with the shoes. So I say there are 23 pair of shoes. Right, now I say how many total shoes are there? 46 shoes are there. So out of those 46, 23 you will wear in your left leg and 23 you will wear in your right leg, right, left foot and right foot. Similarly, out of these 46, 23 is complete one set and these 23 are another set. How many sets of chromosome? 2 sets of chromosome. One, again, just related with your shoe, you will wear in your left foot and the one set you will wear in the right foot, right? But together 46, right? So I can say how many sets are there, students? One set and a second set. How many sets are there? 2 sets are there. So if there are 2 sets, I am going to use the word diploid. Di means 2. So how many sets of chromosomes are there? 2. Since 2, so I am going to represent it as 2n. 2 sets, right? 23 we take from our father and 23 we take it from our mother. 23 from mother, 23 from father. So I can say one set is from father, and the chromosomes which we inherited from the father, we call them as paternal, and the one which we inherit from the mother, those are called as maternal. Maternal, right? So I can say that yes, 23 chromosome, I get it from the mother, 23 from the mother, from the father. Father one are called as the paternal, mother one are called as the maternal. So if the chromosomes number are double, if they are in pair, if they are diploid, we're gonna represent them by 2n. But if we say in gametes, how many chromosomes are there? Only 23. Half, that is only 23. So is it 2 set or 1 set? If I talk about only 1 set, it is just 1 set, 23, or let's say this set again 23. So if I talk about only 1 set, if I am only talking about 23 chromosome, no pair, such type is called as haploid. So haploid means only 1 set of chromosome, N, right? So diploid is nothing but 2 sets of chromosome. There are 2 sets. And if I talk about haploid, they have 1 set of chromosome, right? And haploid will be represented by a single N. Now if I say this 46, obviously they are pair, 23 pair, that is 2 sets. So this will be diploid. This will be diploid. And the gametes have only 1 set. Only 23, only 1 set. So I'm going to call my sperms and egg as haploid. Again the zygote with 46, N plus N, 2N. Is it making sense? This whole concept, is it making sense to you? Now if we look into this pair which is identical, just like I told you, you have 23 pairs of shoes, but if you look into a single pair, that will be identical. Similarly, if we look into these 2 chromosomes, these 2 or next 2, so the paired one, they are almost identical, and the identical chromosomes, they are called as homologous chromosome. So this is a homologous chromosome pair. So what do we mean by homologous chromosomes? So homologous chromosomes are nothing but the identical chromosome. Identical chromosome. Identical chromosome. Now how they are identical? With respect to the size, with respect to the position of the gene. Let's say in this, a gene will be present here. So in the other chromosome, the gene will be present at the same time. Right? So I can say with respect to the size, position of gene, and position of centromere. What is centromere? This main middle part, right? This is the centromere, the one where the 2 arms are attached, right? I hope you all are clear with it. I hope it is making sense to you. Also, if you are not very basically clear about what do you mean by a gene, let me give you a recap of it, just so that you are very much done with it, right? So if I look into a cell has a nucleus, right? A cell has a nucleus. Within the cell, we have a nucleus, and within the nucleus, we know that the genetic material DNA is present in the form of chromatin. What is chromatin? An entangled mass of thread-like structure. This is nothing but DNA only, na? So this is the cell, this is the nucleus, and within the nucleus, we have an entangled mass of thread-like structure called as chromatin. Now, once a cell divides, once a cell is dividing, when a cell divides, so whenever a cell is dividing, this chromatin undergoes more compaction. It undergoes more compaction so as to form a rod-like structure called as a chromosome. it will form a rod-like structure called as chromosome. So the chromosome has these two arms which are attached like these. These two arms are called as chromatids, and the region where they are attached, this is called as centromere. This is centromere. These two are chromatids present in a chromosome. So chromosome we can only see when the cell is dividing, right? So imagine a chromosome as a book, and just like a book has various chapters in it, similarly a chromosome will have various genes in it. So the chromosome will have various genes in it. Genes, not the gene which you are wearing, obviously GENE, right? So these genes are present within the chromosome. Just like if you look into your book, and book has various chapters, so the chapter contains the basic information about it. Similarly, the gene has basic information. Now, what are those information? So from the gene, we can make up a protein. From the gene, a protein can be made, and these proteins, they then control the character. Now, ma'am, what is this now? Let me explain it to you. So, for example, if you talk about a height, either maybe a tall or a dwarf baby plant, right? Or maybe if I say flower color, if I say the seed of the shape, these are nothing but character. But who is controlling? Who is controlling the height of the plant? Who is controlling the flower color? Who is controlling the shape of the seed? These all characters are because there is a certain protein made which is giving that character to it. For example, there will be a protein which is making a plant tall. There will be a protein which will be responsible for the purple color of the flower. So any character is because of a protein which is made, and these proteins, they are made by the gene. So if you can see any character is there, that character is possible because of the gene present, right? And gene is present over the chromosome, right? What is this chromosome made up of? Ma'am, if chromatin is made up of DNA, if chromatin is made up of DNA, obviously the chromosome will also made of DNA. The chromosome will also be made up of DNA. Just imagine there is a book. Book is made up of pages. Book is made up of pages. Similarly, the chromosome is made up of DNA, right? Now I say, tell me about chapter 2. You'll be like, okay ma'am, chapter 2 is from the page 10 to page 25. So you are telling about a pages only, a segment of the book, a segment of those pages, not the entire page. Just imagine this is your chromosome, this is your DNA, right? A segment of it. Let's say this is the segment. A segment of the complete DNA is called as the gene. So I can say gene is nothing but a segment of DNA, right? Making sense? Though this is all you have covered in your class 9, but since in this chapter we're going to use various terms like genes and all, that is why I am discussing about it, right? Coming back to this question that why do children resemble their parent? So I can say this is because the genes, they are transmitted from one generation to another. So we saw the genes are transmitted. How they are transmitted? They are transmitted during sexual reproduction. So yes, once these chromosomes are transferred, what are these 23? These 23 were nothing but chromosomes only, na? So that is, we get equal amount of DNA from the parents, but also since through the genes, right? Genes, they are transmitted to the child through the, and where are these genes present? Ma'am, these genes are present within the chromosomes, and chromosome is present within the cell. And which cell? Basically, in this case, we are talking about the gametes, since gametes are moving from parents to the baby, right? So these are transmitted to the child through the gametes during sexual reproduction. Is it clear? Yes or no? Right? Making sense to everyone, right? Okay. Now one more thing. If I say, let's say these one set you get it from mother, this one set you get it from your father, right? So if I say you get one set from your mother, and obviously these chromosome contain what? Chromosome contain, ma'am, genes, and genes control the character. So I can say for every character you get the genes from your mother as well as from your father, right? So I can mention it that each trait is influenced by both maternal and paternal DNA, right? Let us say you have black eyes. This is because, let's say, your mother is also having a black eye, your father is also having the black eye. So if you're having any character, maybe the black eye, or maybe your skin color, maybe your hair color, any character which you have, that is because of your father as well as your mother. So your mother and father characters are because of the DNA, and your characters are also because of the DNA, half from your mother, half from your father. That was with respect to it. Now you'll be like, sometimes ma'am you're using this word trait and sometimes you're using this word character. What is the difference between the two? So character is nothing but the feature of an organism, the height, the flower color, seed shape, right? But trait is like the alternative form of a character. Let's say you have a box of crayon and I say, please give me a color. You'll be like, which color? So color is the character, and if I specifically say red color, white color, that is a trait, right? The alternative forms of a character is trait, right? If we are being specific, I say, give me a flower color, right? Ma'am, which flower color do you need? Which flower do you need? Then I'll be specific. I need a red flower color. Then you'll be like drawing the color with the red in color or white in color or whatever, right? So when you are describing about that character, right, that will be the trait. So for example, I can write it here. I can write an example here. For example, character is height. Character is a feature like height. And if I talk about trait, trait is the alternative form. Let's say trait will be either the tall or the dwarf. So this is the basic difference between the two, right? Making sense? Chalo, let's move ahead. Now there are various terms which are given in your books, but we'll be understanding these terms first, then we'll be moving forward, right? So just now I told you that there are characters. Characters like, ma'am, characters like, example, flower color, plant height. These all are nothing but the character, the feature of an organism. Now, who control characters? Ma'am, gene control characters. Yes. Perfect, right? So these characters, they are controlled by genes. So gene control characters. We all know this by now. Is it clear? We all know this, right? Now if I say the plant height, let us focus plant height, right? Let us focus plant height in detail. So if I talk about the plant height can be tall or dwarf, right? The plant height can be tall or dwarf, isn't it? Do you agree with me? What are these? The tall and dwarf, ma'am, these are, yes, ma'am, these are the traits. Very good, guys. I hope you're able to relate it, right? So these are the traits. Traits are nothing but the alternative varieties of a character, right? If you want, you can write it here. Alternative varieties of character. Right? So what are the alternative varieties called as the traits? So the trait can be either tall or dwarf. Now who is controlling these traits? Okay, gene will be controlling the height, but what about this? Isn't it little bit confusion? Let me help you with it. Let me help you with it by drawing a diagram and helping you to understand this. Right? For example, this is a chromosome. I'll be like, ma'am, till now you have shown chromosome like this. Yes, a chromosome will be like this. For simplicity purpose, I have just drawn like this. Right? For the simplicity purpose, I'll be drawing like this. So do not get confused. Let us say, ma'am, okay, we are clear till here, right? Great. So, okay, students, this is my chromosome. So, on the chromosome, what is there? Yes, on the chromosome, we have gene. Very good, right? On the chromosome, we have gene. Now, gene make up the protein. Gene is nothing but a, yes, gene is nothing but a segment of DNA. It is nothing but a DNA segment. Now, this gene make up the protein, right? The gene is responsible for the synthesis of the protein. Now, this protein is acting like an enzyme, right? You know what are enzymes? Enzymes are nothing but some substances which increases the rate of the reaction. It is increasing the reaction rate. It is increasing the speed of the reaction, right? So basically this protein is acting like an enzyme. So it is enzymatic in nature, and this enzymatic protein, it is acting very efficiently, right? Its efficiency is quite good. The efficiency of this enzyme is quite good. Its efficiency means it's working very great. For example, right now I am all fit and fine, so I'll be functioning well. I'll be teaching you with utmost of enthusiasm and with full josh, and you are also studying here, right? So this protein right now it's functioning very well. It's all good. So now what is the function of this enzyme? What it is doing? It is helping in the formation of a growth hormone. It is helping in the formation of a growth hormone. That is, it will be causing growth in the plant. What is this growth hormone doing? The name itself says that it will be causing the growth in the plant. So if the plant will be growing, that is, its height will be growing. So if the height of the plant will be increased, that is, the plant will become tall. Yes, I can say if the growth hormone is there, it will be formed in large amounts since we have good enzyme working efficiently, right? As a result, the plant height will be increased, and since the plant height increased, we can say the plant will be tall. The plant height will be tall. Clear? Is it clear? Right? Now let us say that we have another, this is the chromosome, and on this gene there is some changes happening. Some change has happened because of XYZ reason, that this gene is little bit changed, right? This gene is a little bit changed, so therefore I am drawing a star here to signify that this gene is now changed, right? This gene is now changed. Some change is there. Now, since this gene is changed, yes, protein will be made, but it will not be as efficient as it was earlier. The enzyme efficiency will be decreases. The efficiency of the enzyme will be decreases. That is, it will not function as good as it was doing before. For example, guys, now I am suffering from fever. I have headache. I have cold. I have cough. Will I have the same energy? No. Will you, if you have fever, you have cough, right? Will you have same energy? No, ma'am, we will be dull. Similarly, can this protein? Yes, the enzyme will be there. The same enzyme will be there. Just efficiency will be less. It's the same gene, just little bit changes. It's still you. Even if I get a cold, cough, it's still astha only, na? It is not changed. I'm not gonna change. I'm not become pasta. right? I'm making my own fun here. But what I want to tell you that I am not going to change. ASA will remain ASA only, right? So if this protein is there and the enzyme efficiency is decreased, so I can say what will happen? The growth hormone will be there, but the amount will decrease. It will still make growth, right? I will still be able to teach, but will I be able to cover as many syllabus as I will be covering when I am fit? The answer is no, right? So I can say the growth hormone amount will be decreased since it is not functioning efficiently. And yes, if I say amount will be less, the plant height will not be increasing as such. So the plant height will not be increasing as such, and as a result the plant will remain a small heighted plant, that is dwarf. What do you mean by dwarf? Small height, right? The height will be small. A small heighted plant is called as the dwarf plant. Did you see that there are different traits? This is a tall trait, this is a dwarf trait. We have different traits because of, are these two different genes? Astha well and Astha not well. Are these two different people? No, they are same people. Similarly, these are not two different genes, but just different form of a same gene. Just the different form of the same genes. Hence, we can call them as, these two can be called as alleles. These two can be called as alleles. What are alleles? I can say different forms, different alternative forms of the same gene. Different alternative forms of the same gene. Making sense, yes or no? What do we mean by alleles? Is it clear? Coming back to this question, so I can say, if the gene is controlling the character, who controls the trait? The trait is controlled by alleles, right? So I can say gene is having the alleles. The alternative form of the gene is called as the allele. This is also allele and this is also allele. How to distinguish them, right? Astha well and Astha not well. How I'm going to distinguish these two alleles, right? How I'm going to distinguish? So I'm going to give them two names, that is the dominant allele, the one which is functioning very well, right? But the one which is not functioning very well is the recessive allele. So dominant and recessive allele. Dominant and recessive allele. The one which function well, dominant. The one which not function well is recessive. Just relate to them of a jungle. Now, how jungle? So imagine in a jungle there is a tiger and there is a cat. So obviously, who is going to rule the jungle? The tiger will rule, or let's say in place of tiger, lion will rule. Anything can work, right? So tiger will rule even the cat is present or not. Even if the cat is there or not. But can a cat rule when the tiger is there? Cat will be like, no, I'm scared. So cat can only rule if tiger is not there. The cat can only rule if the tiger is not there. So what I want to tell you, that the dominant one is always expressing and the recessive one only express when this dominant one is not present. What do you mean by it, ma'am? I'll tell you slowly and slowly, it will make more sense to you. First, we're going to write this, that dominant one, it always expressed and the recessive one will express only in the absence of dominant allele, right? Making sense? It will make more sense slowly and slowly once we'll move forward, guys. So if I talk about this stall trait, right? These are controlled by alleles. We have seen it. Traits are controlled by alleles. So there are two alleles, right? One will be dominant, one will be recessive. So remember, always the dominant will be expressed in capital letter. And the recessive one will be expressed in small alphabetical letter. Small alphabetical letter. Right? So if I see here, I say clearly the one who is functioning well, this one is dominant, and the one who is not functioning well, this one is recessive. So similarly, this trait, I can, how to distinguish between the two traits? I can say if this is the dominant allele which is functioning well, then I can say this is the dominant trait. If this is the recessive allele which is not functioning so well, this is the recessive trait. Right? So I can say dominant allele controls the dominant trait, recessive allele is there which is for the recessive trait, right? Making sense or not? So I can say which one is recessive, tall or dwarf? Ma'am, dwarf is recessive and tall is dominant. Very good. But if we talk about the alleles, right, so I can say which one will be recessive, how to represent it? So I am going to take the trait of that particular allele. So this allele controls the tall trait. So I am going to take only the capital letter of the first, I should say the first letter of this, that is T, right? What is the first letter of this dominant trait? T, right? I'm going to write it in capital. For the dwarf, I'm going to write it small t. I'll be like, can I write, ma'am, D? No, it will be wrong. Just the small alphabetical letter of the one. Why we are not writing like that? Because you yourself are gonna get confused that, ma'am, D is for what? D is for Don, D is for double, or D is for dwarf. You're going to get confused. So if you have said that, okay, from the tall, that is the dominant trait, we have taken the first letter, that is T, and if it is dominant, I'm going to write it in capital. If it is recessive, I'm going to write it in small, right? This is the dominant allele and this one is the recessive allele. Making sense? Yes or no? Yes, right? Now guys, in humans or in generally in organism, in plants also, the chromosome occur in pair. Just like your shoes occur in pair. I told you this few moments back, right? So that is what that chromosome occur in pair. Chromosome, they occur in pair, isn't it? That yes, ma'am, we have like this. Okay, maybe we can take this topic on the next page so that it will make more sense to you, right? So we can say chromosomes, they are paired, right? That is, we have a diploid situation. We have a diploid situation. We have two sets of chromosome, right? Suppose this is the chromosome. Again, as I told you, chromosome has two chromatids. For simplicity, I'll be only drawing one. And therefore, it will have its pair. Every chromosome will have its pair. What we saw that on the chromosome, what will be there? On the chromosome, alleles will be there. On the chromosome, alleles will be there. So, ma'am, which allele? Either dominant or recessive. So, yes, let's say here an allele is there. Either capital T or small t, whatever can be the possibility. And here on the same position, I can say, yes, a capital T or a small t. Since the chromosomes are in pair, these are the homologous chromosomes. Since the two chromosomes which are identical, right? These are homologous chromosomes. Since I say chromosome occur in pair, therefore the alleles on them will also be paired. Where are alleles present? Alleles are present on chromosomes only. Since chromosomes are paired, we can say alleles are also paired. So what will be the combination? Either capital T will be present or a small t, either capital T or small t. So I can say total 3 combinations can be possible. So I can say, we can say 3 combinations can be possible. That is, what are these 3 combinations? So either it can be capital T, or maybe on both of them small t, or on one capital T and on the other small t. These are the three combinations possible. Either both capital T, either both small t, or one capital T or one small t. You will be like, one situation, ma'am, can be small t and capital T. Yes, but this is nothing but this only. This is nothing but this situation only. But remember, always we're going to write the capital letter first, followed by the small letter. So this will be preferably written like this. But this and this are actually the same thing. But we're going to write it like this. So three situations are possible. Okay, ma'am, if capital T is there, we know it will be tall. So if both the capital T will be there, it will be giving rise to a tall plant or a dwarf plant. If both capital T, obviously it will be tall. Nothing fancy about it. Both small t, okay, small t is responsible for which trait? Small t is responsible for dwarf, capital T for tall. So obviously if it is only small t, I can say it will be dwarf. Right? Just relate with it. For example, I told you if tiger is there, tiger is gonna rule. Both the tigers are there. Who is gonna rule? Tiger is gonna rule. Right? Only cat is there. Cat can only express in the absence of tiger. So will this small t express? Yes, because tiger is not there, capital T is not there, the dominant one is not there. So the small t recessive one will express and give rise to a dwarf plant, a small heighted plant. And again this one will be tall or dwarf. Again a tiger is there and cat is also there. If tiger is also there, cat is also there, who is gonna rule? Tiger is gonna rule. Cat will be like, okay. So tiger will rule, that is capital T will express, and capital T is responsible for, yes, that basically the dominant one will rule, and the dominant capital T is responsible for the tall trait. So it will be again tall. Clear? So three conditions which were there, if I look into these three combinations, I see that these two have the same alleles. If on both the chromosome the alleles are same, we call such situation as homozygous condition. Homo means same. If on both the chromosomes we have two same alleles present, either capital T or small t, right? And if I say two different alleles are present, then I can call it as heterozygous condition. Heterozygous, hetero means different. I can say if two different alleles are present, that is one capital T and one small t. Making sense, yes or no? Okay. Now, in the homozygous, we have two situations. If both are dominant, right? Both are similar and the similar one is dominant. So I can call it as the homozygous dominant. This one is homozygous dominant. And if both are same but both of them are same and both of them are recessive, I can call this situation as homozygous recessive. Homozygous recessive. Making sense? Yes. Clear? Are you able to relate with these terms, right? So if I look into this physical appearance, either it is tall or dwarf or maybe violet or white, all these are nothing but, yes, the physical feature. So the physical feature can be called as the phenotype. Phenotype. Phenotype means Phenotype means physical feature of the organism. Physical or external feature of organism. But if I look into what type of genes are present, what type of genes are there, that tells us about the genotype. So these three, capital T, small t, capital T, small t, these are nothing but the genotype. Genotype is nothing but the genetic makeup or genetic constitution of an organism, right? So I can maybe mention here genetic makeup or genetic constitution of an organism, it is called as the genotype. This is called as the genotype. Making sense to all? Right? All these terms are clear, yes or no? Yes, all done, right? Now we move further. So if we move further, we have these terms, right, which are mentioned here. What do we mean by factors? Factors are nothing but genes. So Mendel used the term factor or unit factor for gene. So gene is nothing but unit factor or factor, right? We have seen this term alleles. These are just for your definitions. I have already explained you the terms, right? That is why I didn't directly go with these terms, because if I have told you just by the definition, you must have not understood it, right? That is why I have explained you first. Now we are coming to definition, so that if the question comes to write these definition, you can. So it's very rare that such kind of question comes, but yeah. So alleles basically are nothing but the alternative form of a gene, right? Then we move to homozygous. Homozygous once both the alleles are same, that is either capital T, small d, small t. Heterozygous once the two alleles are different, right? For example, here. Making sense? Yeah. Again, phenotype, we know the physical feature, the external feature, right? And genotype is the genetic constitution. So that were about some of the terms. Let's move ahead. So we have seen what is genetics and who is the father of genetics, the one who experimented for 7 years on a plant, and he performed various hybridization experiments on that plant. So what was that plant? So who is this person? This person is none other than Mendel. So Mendel is the father of genetics, and he performed his experiments on garden pea, or also called as edible pea, or also the scientific name is Pisum sativum, right? On the pea plant, right? We call it as the garden pea or the edible pea, right? Now if you write the scientific name, please write the P capital of Pisum, please write S small of sativum, and underline them separately. Underline them separately. This is the scientific name of garden pea. Now he experimented on the garden pea for 7 years. What was so special about it? Why Mendel selected garden pea for his experiment? A very, very important question. Because this plant was a short plant, right? We can easily use it. Also, the lifespan is short. It has a shorter lifespan, right? So I can say it has a short lifespan, that is of only 3 to 4 months. What is the benefit of having a shorter lifespan? So I can say that we can obtain the results for very quickly. We don't have to wait for years and years to see the result. Is it going to be tall or dwarf? It's going to be very quick, right? So I can say results of the experiments were obtained in the less time. Next, we can say it could be grown throughout the year. Like, it's not like we have to wait for a specific time to grow them, right? In a year. Right? Next, choice of cross or self-fertilization. Since it is a bisexual flower, you know we have studied this in the last chapter. That is how do organism reproduce. That is, it has the male part as well as the female part. So easily what can happen? Self-pollination can easily happen, but also we can perform cross-pollination artificially if we remove the male part. If we remove the male part. So we can easily conduct the experiment. That is self or cross. Remember, cross one artificially. We are performing cross pollination artificially. Lastly, yes, we have 7 different types of character. That is, he chose different types of character, and he can perform different experiments on different types of characters, right? So yeah, that was about it. And also the large number of offspring, it will give rise to large number of babies. What is the benefit of it? Let's just say if I given a assignment to 2 students, and the same assignment, for example, I'm asking you, do you study during the daytime or do you study during the night time? And I have to make a report of it. So if I'm asking just 2 students, and on the other hand I am asking 200 students, from where my report will be more reliable and better? Obviously where I'm collected from the set of 200 or 100 students. So basically more number of offspring means a good number of data for the statistical analysis. Right? Is it clear why we selected garden P? What are the properties? What are the advantages? Right? Now I told you Mendel selected 7 character. 7 character. Now each character is having 2 pairs. That is, each character has 2 traits. Right? One character has 2 traits. Then 7 character have how many traits? 7 into 2. 14 traits total, right? 14 total traits. One more thing, remember guys, please add it here only, that the homozygous one is also called as the pure line. If you ever see the word pure line or true breed, remember they are talking about the homozygous condition. And if you see this word heterozygous, there will be word called as impure or hybrid. Another name for the heterozygous is impure or hybrid, and another name for homozygous is pure line or true. Coming back to the topic where we were. So he selected 7 characters. Let us see all the 7 characters. The seed shape and the seed color. The very first thing, the seed shape and the seed color. So the seed shape can either be round or wrinkled, right? Seed color can either be yellow or green, right? So on this side, this is the trait mentioned, and on the very first column, what they have mentioned? The character, right? So seed shape having 2 character, round or wrinkled, seed color yellow or green, right? Then we come to flower color. Flower color can either be violet or white. Then flower position can be axial or terminal. What do we mean by it? So if we have a flower at the tip, at the apex means, right? Apex means at the tip, at the terminal side, at the end, right? Terminal means apex, apical. So if it is at the tip, then we call it as a terminal position. But if it as the axial means this is the stem, this is the leaf, right? If between this, the angle between the stem and the leaf, if flower is present between the two, we call such position as axial position. Could you see? Axial position, and here the flower is at the tip, apical position. Lastly, we move to the pod shape and pod color. So the pod shape can be either full, right? A flat appearance will be there. But if it is having this beaded appearance, we call it as the constricted, right? Full means it will be having a flat appearance, and beaded appearance means constricted, right? Next, we move to pod color, either a green or a yellow. And lastly, we have the stem height, which you already know, tall and dwarf. Now, why I am telling you? Because in NCERT this table is not given. But ma'am, do we have to study it? Yes, very important, because in exams they're going to give you different questions based on this table. So do not forget to remember it. Now you will be like, ma'am, we already know that tall is dominant and dwarf is recessive, but what about the other 6 characters? So the ones which are mentioned on this side, these are dominant, and the one which are mentioned on this side, they are recessive, right? Dominant and recessive trait. So that was about it. Now how Mendel performed his experiment and what all he did, let's discuss about that now. Okay. Alright, let's move to the next topic, that is how Mendel performed his experiment. What is the technique? The technique is simple. The Mendel took the parents, he selected the parents, then made a cross between them, and then again made a cross between the F1. Be like, what is all this, ma'am? So I'll be like, wait a minute, give me some time and you will be able to relate. The very first thing is the mono-hybrid cross. Definitely a 4-marker question, a case-based question definitely comes from this particular topic, mono-hybrid cross. So mono-hybrid cross is a cross where we are taking two individuals which are differing only in one pair of contrasting trait. One pair of trait means two traits. Two traits means one character. That is, we are taking two plants which are differing only in one character. Two plants differing only in one character. So what we did here? We took the parents. I told you Mendel selected the parents. We're going to write the parental generation here. What will be the parental generation? Mendel took tall and dwarf plant, and both of them he took in the homozygous condition. What is homozygous condition for tall? Tall is a dominant trait, so we're going to write capital T and capital T. We have to write the homozygous condition, not the hetero, right? Parents will always be in the homozygous condition until and unless mentioned otherwise, right? So tall, dominant, capital T, dwarf, homozygous, small t. Nothing scary about it. Then what we are going to do? We are going to, just like it's a sexual reproduction. I told you in sexual reproduction there are two parents, okay? What is the second step? The second important point about sexual reproduction is there is involvement of gametes. Obviously, first we're going to make the gametes. So if I draw the gametes here, I told you, right? Let's go back to this page. Let me just remind you here. So I told you in gametes only one set is there. So I told you since the chromosomes are paired, the alleles are also paired. We discussed it. Since the chromosomes are paired, the alleles are also paired. But now these are not going to be together, right? Out of the two set, only one set will be present in the gamete. The gametes only have 23 chromosomes. Only one set will be there. So I can say since the chromosomes are not together, these paired alleles are also going to separate. Isn't it? Out of the two, only one will be present. So I can say out of the paired one, only one will be there in the dihybrid cross. So basically, I can say the gametes will have out of the two, only one. Now what will happen? They are going to cross-pollinate. Cross-pollination will be there. Obviously, what will be the offsprings like? What will be the babies like? Capital T and small t together. Now it is a tall or dwarf. Obviously, since we have a dominant allele here, it will express and give rise to a tall. Now capital T, small t is the genotype or phenotype. It is the, yes, genotype, and tall is nothing but the phenotype. Very, very good. Now this one which we have found, this one is called as the F1 generation. F1 generation means first filial generation. First filial generation. That is the generation, the offsprings, the progeny which are formed after the cross between the parents. This one is the first filial generation, F1 generation. Right? What is the second step? So I can say all the plants which are formed, all of them are going to be like this. Now what we're going to do? We're going to self-pollinate. What we're going to do? We're going to self-pollinate. What we're going to do? We're going to cross F1 into F1. We are going to make the selfing. Selfing is also called as the self-pollination. Self-pollination. That is what we're going to do. What is the F1? F1 is capital T small t into F1 capital T small t. The same step will be there. First we will be making the gametes. Very good. First we will make the gametes. What will be the gametes now? Okay, I told you this pair will not remain together. This pair separates. Now imagine if I have a birthday and I have decided to distribute maybe dairy milk, right, in the whole class, the normal dairy milk which we have, right? So I have tried to distribute that chocolate in the class, right? But since I have only dairy milk, all the students will get only dairy milk. But there is my friend who has brought some dairy milk and a few, maybe let's say Bournville chocolate. Okay, Bournville and dairy milk, two types of chocolates are there. So my friend has two choices. So she can give to 50% students dairy milk and to the other 50 students the Bournville, right? So I can say only if you have a choice. Here only capital T was present. Obviously out of the two only one will be there. Out of the two in a single gamete only one allele will be there, not pair. So a gamete will get only once. But since we have only one type of allele here now, but here we have two different alleles. What will happen now? So the two types of gametes will be formed. Yes, they will have only single allele, but one type of gamete will have capital T and one type of gamete will have small t. So how many gametes are formed? Two types. Here also we can say two types of gametes are going to form. Capital T and small t. Now we're going to make a cross between them and we're going to show it here. Now to make a cross between them, we are going to use a Punnett square. We are going to make a Punnett square. Now what is Punnett square? Punnett square is the one which was given by Mr. Punnett. Now what is this square? We are using to find our F2 generation. We are using the Punnett square to find our F2 generation. So what we are going to do, let me just make this table for you guys, then we will write, okay? Seconds, then we will be writing in it. Hello, let's write it down. Okay, so what I'm going to do, I'm going to write two gametes, first two gametes in the row, the other two gametes in the column, right? First two gametes in the row, other two gametes in the column. And now I'm going to put or fill in the boxes. Just remember wherever you want to fill. Suppose this is where I want to fill. Just go vertically upwards and horizontally towards the left, and whatever is the combination right here. Vertically up, horizontally left, whatever is the combination right there, right? So let me fill it for you. So I'm going vertically up, horizontally left, I'm going to get capital T. Here, vertically up, small t, horizontally left, capital T, right? Here, vertically up, capital T, horizontally left, small t. Again, vertically up, horizontally left. So how many? These four which are formed, these are nothing but your F2 generation. The generation which is formed by the cross between the parents is called as the F1. The generation which is formed by the cross between the F1 is called as the F2 generation. So these are my F2 generation. How many types are there? 1, 2, 3, 4. What will be the phenotype? Tall, tall again. This one, tall, and this one will be dwarf. Clear? Clear? So if I ask you what is the phenotypic ratio? Phenotypic as in the phenotype ratio. So you will be saying, okay, how many phenotypes are there? Either a tall is there or a dwarf is there. Two types are there. Either a tall one is there or the dwarf one is there. How many tall plants are there? 1, 2, 3. How many dwarf plant is there? 1. So 3:1. 3:1. Or if it is asked to write the genotypic ratio, to genotypic ratio, the genotype ratio, then how many genotypes are there? Capital T is there. Then we have capital T, small t. And then we have small t, small t. How many capital T, capital T? Only one. How many capital T, small t? 2. And how many small t? Only 1. So is it clear? The phenotypic ratio of the F2 generation is 3:1 and the genotypic ratio of the F2 generation is 1:2:1. Is it clear? So you can, for example, if I ask you in the F2 generation, what is the proportion of tall? If the question is like proportion of tall in F2, right? If the question is proportion of tall in F2, you'll be like, ma'am, okay, out of total 4, out of 3 plus 1, 4, out of total 4, how many are tall? 3. So 3/4 is the proportion, right? If they ask proportion of dwarf, 1/4, right? If in this question, if they ask proportion of capital T, small t in F2, what are you going to write? Okay, ma'am, out of 2 plus 1 plus 1, how many? 4 again. Out of 4, how many are capital T, small t, ma'am? 2 are there. So 2/4, that gives us 1/2. Half are capital T, small t. If they ask you the percentage of capital T, small t in F2, what are you going to do? Just multiply by 100. Just multiply by 100. What are you going to get? 50%. I hope you're getting, you can use your maths here, right? So Mendel used maths in his cross. So basically here also I can say this is nothing but the 75%, this is 25%, 25, 50 and 25 again, right? 25 plus 25, 50 and 50, 100. Right? So you can use your maths here and try to do these questions because these questions can be asked. Such type of question, I just gave you few examples, right? So yeah, you don't have to write it here. This whole will be the part of your cross. The monohybrid cross is this much. Okay, this is the F2, this is the F1 phenotypic genotypic ratio. So what we observed in the monohybrid cross that we have already seen. So lastly, we'll come to here that you saw that in the F1, out of the 2, only dominant trait can be seen. Out of the 2, only dominant trait can be seen in the F1, but in the F2 we saw dominant as well as recessive trait in the ratio 3:1. So here we couldn't see dwarf trait, but here the dwarf trait reappeared, right? So we can mention here that ratio of the tall and dwarf plant was 3:1 and the dwarfness which was not visible in the F1, it reappeared in the F2 generation. Rest all the crosses mentioned which I have already discussed with you. Try to read it and it will mean the same to you which we have already discussed, right? Let's move ahead. Okay, this is the why I told you that diagram was important. Which diagram I'm talking about? I'm talking about this table, right, where we discuss about the different character. Why I told you this one is important? Because in your exams, different types of crosses can be asked. What they have done, they have taken a violet flower. A violet. For violet, what I am going to write? For violet, what I am going to write? Capital V, capital V. Again, a white color flower will be there. Again, as I told you, we're going to take the homozygous condition. So what we're going to write? Small v and small v. We made gametes and ultimately we made the F1 generation. The gametes will be capital V, here small v. Let me write it for you. Capital V, small v. If we fuse them, we get the F1 generation, which is capital V and small v. Clearly it is violet. Clearly it is violet. Then we make a cross between F1 into F1. F1 into F1, that is capital V small v cross capital V small v. What will be the gametes here, ma'am? Capital V small v. These two won't be together. And if you make a Punnett square between these, we can make a quick Punnett square here for you, right? So that you can practice from here itself. So we have added these two gametes here, these two gametes here. And then we're going to make a cross and we're going to write it. Let me write it for you. So we could see out of these four, these three are violet. Out of these four, these three, they are violet. And the last one is white. So we saw always, remember in monohybrid cross, maybe you take about the seed shape, the round and the wrinkled, or maybe you talk about the flower position. Any character, any trait, remember always, always in the F1 dominant trait. Always is the F1 dominant trait. All the progenies will be produced, will be dominant in the heterozygous situation. In the heterozygous situation. Heterozygous situation. And remember, in the F2, we're going to find it in the ratio 3:1. The dominant is to recessive trait. We're going to find it in the 3:1. And remember, always, the genotypic ratio will be 1:2:1, where 1 will be homozygous dominant, 2 will be heterozygous, and the 1 will be heterozygous recessive. This is fixed. Remember it, right? Try to do it. This is my homework from my side to you guys. We have done for the tall, we have done for the violet color and white. Try to do it for the others one as well, right? Let's move ahead. So now Mendel, what he concluded with the help of the monohybrid cross he made. So he actually gave 3 postulates, right, using the results of the monohybrid cross. The very first one is principle of paired factors. Now guys, since we have already discussed so much, we have learned so many terms, it will be a smooth ride for you now. So I am saying smooth ride, you're going to understand it very soon. Principle of paired factors. I told you that Mendel called the genes as factors or the unit factors, and I told you these genes, these alleles are going to be paired. Why? Because our chromosomes are paired. We are diploid organisms, so we know these chromosomes are paired, hence the alleles are going to be paired, or shall I say these unit factors are going to be paired. The pairing can be in such a way that both of them can be same, either capital T or small t, or they can be different, one capital T and one small t, right? This is what it is mentioned here, that these are controlled by the pair of unit factors and they are occurring in pair, and one can be similar and the one can be dissimilar. Similar means capital T or small t. Dissimilar means capital T, small t. Now, pair of dominance. We see in this, obviously, if both are capital T, no one is dominant. If both is small t, no one is dominant. But if we talk about this dissimilar situation, one is dominant and one is recessive. Capital T dominant, small t recessive, right? So I can say a single individual and 2 different allele is present. Then what happens, that one allele is dominant over the other. That is one allele. Which allele? Capital T is dominant over the other, which is small t, right? You know what is dominant allele is doing? It will express itself, right? And lastly, the law of segregation. The law of segregation, that we see that these unit factors which were paired, these alleles which were paired, they separate during gamete formation. That is, a single gamete will get only one unit factor. The single gamete will get only one unit factor. Okay, clear? Yes or no? One more thing, have you just discussed, observed from here that capital T tall, small t dwarf, capital T small t will be tall again. So did you observe one thing? Let me go back and let me ask you this question. If we observed, the dominant one, it is always expressing, means it will express in both homozygous and heterozygous condition. Oh my God, what have you said, ma'am? I hope you are able to relate, by the way. either it is homozygous condition, capital T, then also it will be dominant tall. Either it's a heterozygous condition, capital T, small t, it will be tall. The dominant will express. The dominant expresses. But the recessive one will only express, the recessive one will only express in the homozygous condition when there are two copies, right? So it express in homozygous condition. Express only in homozygous condition. This is also a point which you can mention here, right? I hope you observed it by yourself. If not, it's totally fine, right? We have told you. Okay, let's go back to where we were discussing. So yes, these were the three postulates. That is the principle of pair factors, the principle of dominance, and the law of segregation. Let's move ahead towards the dihybrid cross. Dihybrid means we'll be talking about two characters at a single time. That is, we are taking two plants which are differing only in two characters. Monohybrid only one character, dihybrid two character, right? So we can say we can take two characters. Two character means, let's say Mendel took the seed shape and seed color. The seed shape and seed color. Two characters, okay. For each character, how many traits do we have? We have two traits for each character. Okay, let me write down for you guys. So we have two traits. So for seed shape, what are the two traits? Okay, either it can be round or it can be wrinkled. And for the seed color, again, either it can be yellow or it can be green. Either it can be yellow or it can be green. Which one is dominant, round or wrinkled? Round. So we can represent it by capital R and wrinkled by small r. Remember, the round one is dominant. This is not showing the increasing side, just to tell you this one is dominant. And for yellow, capital Y, and for green, small y, because the yellow one is dominant over the green one. This is the basic idea. How many total traits? 4 traits. 4 traits mean 2 pairs. 1 pair, 2 pair. And that is why it's mentioned 2 pairs. This is 1 pair, this is second pair of traits. Right? That is 2 characters. Okay, let's continue this. So what? Again, the same process. We're going to take the parents, make a cross between them, get a F1 generation, then make a cross among the F1, get a F2. Simple, easy, right? So let's start again. We have to take our parental generation. What is the parental generation? We have to take the 2 characters at one time. So basically the goal of this cross was to know, is inheritance of one character, is it affecting or is it influencing the inheritance of the other character? That we want to observe, right? So we have to take the parental generation. The parental generation is going to be round and yellow. Round, yellow, and let's say the other parent is going to be wrinkled and green. Wrinkled and green. Now again, for each of it, write down the homozygous condition. So homozygous condition for round will be capital R, capital R. Homozygous for yellow will be capital Y, because both round and yellow are dominant. And we have to write the homozygous condition. Homozygous for wrinkled, small r, small r. Homozygous for green, small y, right? Since both wrinkled and green are recessive. Next step was to make the gametes. Next step was to make the gametes. Now what will be the gametes? Out of the pair, only single one will go. Out of the pair, only single one will be going. So we have only one type of gamete, capital R, capital Y. Here also, out of the pair, only single type of gamete is going. Out of the pair, only single type of gametes will be there. Now what are these two gametes are going to do? They are going to fuse, they are going to cross-pollinate among themselves and form the first generation. What will be the first generation? F1. Combine these two, capital R, small r, capital Y, small y. Do not write like this. This is not the way. Always R with R and Y with Y. Write Y with Y and R with R. So this is the preferable thing. So this and this are the same thing, but preferably we're going to write it like this, right? What this is the genotype, isn't it? All the plants will be obtained will be of this type, that is capital R, small r, capital Y, small y. This is the genotype. If I ask you the phenotype of it, if I ask you the phenotype, it will be, this is round, obviously because capital R is dominant, and this one be yellow. Round and yellow plant will be there, round and yellow seed will be there, right? This is nothing but the phenotype. The physical appearance is phenotype. Phenotype. Clear? The next point is F1 into F1. F1 into F1, that is selfing, that is self-pollination. What is F1? F1 we have capital R, small r, capital Y, small y cross with capital R, small r, capital Y, small y. Next step is to form the gametes. We're going to make the gametes here. Now, how to write it? How to write it? How to make the gametes, right? So basically, remember, these pairs can't go together. These pair cannot go together. These, just remember, the pair cannot go together, right? But obviously, it can go with the other allele. One pair of allele cannot go, but it can go with the other allele, na? Right? Is it making sense? Yes or no? Right? So let's say capital R and small r, they cannot go together. I'm going to write them separately, but this can go with capital Y also. It can go with small y also. Similarly, small r can go with capital Y as well as small y. So what will be the 4 types of gamete? Capital R with capital Y, capital R with small y, small r with capital Y, and small r with small y. These are the 4 types of gametes which are formed. You can mention here capital R, capital Y, capital R, small r, capital Y, and small r, small y. 4 types of gametes. Similarly, here also 4 types of gametes, the same gametes. Right? What we're going to now do, we are going to make a cross between them. We are going to make a cross between them. Right? The F2 generation. F2 generation. So for the F2 generation, we have to write the Punnett square. We'll write this gametes in the row, other gametes in the column. Let us write down. Let us write down. So I have a table already constructed here. So let us write down one type of gamete here. These are the 4 types of gametes, right? Capital R, capital Y, capital R, small r, capital Y, and small r, small y. Same gametes will be here. Now what we're going to do? We're going to write the combination by going vertically up and horizontally left. Let us write it down, guys. Let us write it down. So this will be capital R, capital Y, capital Y, capital R, capital Y, small y, capital R, small r, capital Y, and yeah, we're going to fill it very quickly. Please check if I am facing any difficulty. You also do it by yourself. Don't just copy what I am drawing, okay? Try to do it by yourself and then you check it with me. Check if I have drawn correctly, check if you have drawn correctly or not, right? Okay. Very quickly I'll try to fill it. Just go vertically up and horizontally left. You will be able to get your answer, right? Okay. Write down the phenotype of each one of them. I'm not writing, but what I want to tell you, if I have to look for the phenotypic ratio of the F2, if I have to look for the phenotypic ratio, the phenotype of the F2, I hope you all have written the phenotype also. If not, pause the video, write it down. Okay, write it down and then come here. Just see what parts I am marking. Just observe these 4 and these 2. Observe these 1, 2, 3, 4, 5, 6, 7, 8, 9. Observe these 9. All of them are same. All of them are same. Why I am saying same? All of them are round and yellow. Yes, different genotype, but all of them are round and yellow, aren't they? Yes, they are. If I mark these, if I mark this, all of them are round and wrinkled. Check them. All 3 of them. All 3 of them are round and wrinkled, isn't it? Okay. And if I mark these 3, let me use another color maybe. These 3. These are wrinkled and yellow. These 3 are wrinkled and yellow. And lastly, we have this one. This one single is wrinkled and green. The single one is wrinkled and green. Isn't it? Let me just check it once. We have not made any mistakes here. Just let me check. You also check by yourself. Capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, capital R, capital Y, small y, capital R, y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y, capital R, small r, capital Y, capital R, small r, capital Y, yes, small r, capital Y, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y, small r, capital Y, small y. I guess I have done correctly. I hope you have also done it correctly and you have checked with this table also, and you must have found the same results. That is what we are looking at. Out of the total 16, how many are round and yellow? 9. How many are round and wrinkled? 3. How many are wrinkled and yellow? 3. And how many are wrinkled and green? 1. So if we see that out of these 16, these 10, they are parental combination. They are same as that of the parent. Parent was round and yellow and wrinkled and green. Right? In the F1 also only round and yellow was there. But in the F2 we are having some new combination. Right? These 2 aren't these the same as your parental generation? Aren't these combinations your parental combination? The same one, round, yellow, wrinkled, green. But these 2 were neither present in your parental generation or in the F1. These are something new. They are new combination. And these new combinations, they are called as recombinants. Recombinants. Some new combination is there. And why this happened? Recombinants. And why this happened? Remember, in the starting only I told you that why we are doing this cross, because we want to check whether one character is interfering, is influencing the inheritance of the other character. Is it such a case that this seed shape is restricted with this seed color, or this seed shape is restricted with this seed color only? What I mean to say, is it like that, for example, you are there, Ram is there, and Ram want to sit with Sham. Sham is the best friend of Ram, but it's not like if Sham is absent one day, Ram is going to sit alone. Ram is like, I am not gonna sit with anyone, I just want to sit with Sham only. No, Ram is like, okay, if Sham is not there, maybe I can sit with Rama or Sham or whatever names, the Gita, Sita, whatever, right? So Ram is not restricted that Ram is gonna sit only with the Sham. Ram can sit with Gita, Rita, Mita, whatever. Similarly, it's not like the round will only be there with the yellow and wrinkled will only be there with the green. If would have happened, we would not have find these new combinations. We would not have find this new combination. So it tells us that one character is not influencing the inheritance of the other character, and two traits will, or shall I say the pair of the traits, they are going to inherit independently. The two characters inherit independently to each other. They are segregating independently. That is what we got from the dihybrid cross. That is the law of independent assortment. What do I mean by that? That these alleles of two different characters, they segregate. They are assorting independently. It will not be interfering with the other one. These two are totally independent. Okay, that was about law of independent assortment. Remember, if the question comes what Mendel got from it, what was the conclusion of the dihybrid cross, it is this: independent assortment is not from the monohybrid cross. Remember it, we are getting independent assortment from the dihybrid cross. From the monohybrid we were having those three, right? The pair, factor, dominance and segregation were from the monohybrid, but this independent assortment is truly and truly from your dihybrid cross, right? That was about it. Let's move ahead. How do traits get expressed? We have already done it, right? Where we did it, this was nothing but how the traits get expressed. Let me just show it to you. This was nothing but how the traits get expressed. We have already covered it, right? But the theory part is mentioned. You can have a look at it. This was the question and this is the answer to this question, right? You already know the main part. The theory is mentioned, which you can have a look from there, right? Where we were? Yes, right? Just have a look at the theory, go through it. You will be able to relate, though we have discussed it. Moving forward towards the sex determination. Right? Now, what is sex determination? Sex determination is a mechanism by which we are determining the sex of the individual, whether it will be a boy or a girl, right? So, we can say mechanism by which sex of the individual can be determined. Sex of the individual can be determined. That is whether it will be a boy or a girl. That is what we are determining. Now, if we talk about humans, how they determine sex of the individual. So basically, different organisms have different strategies to determine the sex. For humans, we determine the sex with the help of the genes, that is genetically. So with the help of the chromosomes, right? So I can say this human beings, the sex is determined genetically. Sex is determined genetically, that is with the help of chromosomes. That is with the help of chromosomes. Now, how many chromosomes we have? How many chromosomes are present in a human cell? Okay, the answer is very clear and is known to all. That is 46 chromosomes. Remember, out of these 46 chromosomes, 44 chromosomes, or shall I say how many pairs? 44 chromosomes mean how many pair? 22 pair of them, they are called as autosomes. 22 pairs, they are called as autosomes. And rest of them, they are called as heterosomes, or also called as sex chromosome, or also called as allosome, right? There are other names of this: sex chromosome, heterosomes, allosomes, different names. So 46 out of 46, 44 are autosomes. Then how many are left? Obviously, simple calculation, 2. So 2 chromosomes are called as sex chromosomes. Why they are called as sex chromosome? Because they will be helping in determining the sex of the individual, right? These are involved in determining the sex of the individual, whether it will be a boy or a girl. Now we know we have 46 chromosomes. So out of the 46 chromosome, remember, if we talk about these two sex chromosomes, what are these two sex chromosomes? They have a name, by the way. They have a name, and the name is X and Y. They have a name X and Y. So if we look into our male, so males have two different sex chromosomes, one X and one Y. On the other hand, the females have two same sex chromosomes, that is X and X. Very important point which is mentioned here. Right, very important point. That is, the males have X and Y, two different sex chromosomes, and the female have X and X. Remember, if we talk about the X is slightly normal sized chromosome, and Y is slightly smaller, right? It is slightly smaller in size. This chromosome is slightly smaller in size, the Y one. Right. So basically, yes, if we talk about humans, we have 46 chromosomes. But if we talk about in case of males, only one pair is not perfectly paired. So I can say if it is X and Y, in male, it's not perfectly paired, right? There is one pair of mismatched chromosome. They are not perfectly paired. And if it is as not perfectly paired, which one is that mismatch? That one is X and Y. But in female, all of them are paired, perfectly paired. In females, all of them are perfectly paired. Now, how do we determine the sex? Let us have a look at this particular figure to understand it. So we have taken the body of the male and we have female, right? We have male, we have female. If it is male, it's going to be XY. If it is going to be female, it's going to be X and X, right? The gametes will be formed. Again, the same concept, the gametes will be formed. The gametes of the males are called as the sperm, and the gametes of the female is called as the egg. These two chromosomes will separate, so only one will be there. Only X will be there. So the egg will have only X. But since here these two chromosomes separate, but since they both are different, so 50% of the sperm will have X and 50% of the sperm will have Y. So we can say in case of male, two types of gametes are formed. One having the X chromosome, one having the Y chromosome. Obviously, in males and females, these autosomes remains the same. There is no difference as such. Only difference is there in the sex chromosome. So males have two types of the gametes. Female have only one gamete. So this sperm can fuse with this egg to form the zygote, or maybe this sperm can fuse with this egg to form the zygote. Right? Either it can fuse with this one, either this one can fuse. Whatever can be the chances, right? So if this one fuses with this egg, it will be X and X. And X and X, we all know it is going to be female. But if the sperm containing the Y chromosome fuses with the egg, it will be X and Y. And X and Y is obviously going to be male. So I can say if there is reproduction taking place and the babies are formed, there is a 50-50% chance that a female or a male, or shall I say a girl child or a boy child will be there. But did you see one thing from here? That the mother, this is the mother, right? The mother is giving X chromosome. This mother is giving only X chromosome either to the female, either to the male, either to the girl child, either to the boy child. Mother does not have any choice. It's giving only X. But if the child inherits X chromosome from the father, if the child inherits X chromosome from father, it will become girl. But if the child inherits Y chromosome from the father, it will become boy. So can I say the sex of the child is determined by the type of the chromosome inherited from the father? If child inherits X chromosome, it will be girl. Child inherits Y chromosome, it will be boy. Right? So this is what it is mentioned here. Please have a look into it, that all the children will inherit X chromosome from the mother, regardless boy or a girl, but the sex will be determined by what type of chromosome they are inheriting from the father. Child, if inherits X chromosome, it will be a girl, and the child who inherits Y chromosome from the father will be a boy. That was about the sex determination in humans. But if we talk about the sex can be determined with the help of the environment also, in the case of turtle and lizard, that is how environment, that is with respect to temperature. So at what temperature the eggs are kept, it will determine the sex. Maybe if it is kept at high temperature, it will be a boy or girl. If it is at low, maybe a girl or a boy. Right? So the temperature basically determines at which the eggs are kept. Right? And lastly, we can talk about snails. So snails basically, guys, they change their sex. Snails also, they change their sex, and again, not genetically, right? Change their sex, right? So in case of environmental sex determination, we have seen the lizard as well as the turtle, which can change their temperature, but snails, they change their sex, right? So temperature at which the eggs are kept will determine the sex, that is with the help of the environment. Turtle and lizard changing. Both of them are not genetically determined, obviously. Both of them are not genetically determined. Okay. How Mendel experiments show the traits are inherit independently? We have to draw the monohybrid or dihybrid cross here. Since they are talking about inherit independently, we're going to talk about the dihybrid cross here. Nothing fancy about it. Very interesting. You will be able to do it. Let me move forward. Right. Next question shows us in monohybrid cross what is the ratio of homozygous dominant and homozygous recessive in F2. So if I talk about F2 has, okay, let's talk about tall and dwarf, right? So what is the F2 genotypic? Since they are talking about homozygous dominant and all that, we are talking about the genotypic ratio. The genotypic ratio was 1:2:1. That is capital T, capital T, capital T, small t, and small t, small t. This was the F2 ratio, remember? Now they're talking about homozygous dominant. This one is homozygous dominant. Homozygous recessive. This one is homozygous recessive. What is the ratio? 1:1. 1:1. D is the correct answer. We are taking 120 plants and we are making a cross between a pure red. Pure red means they are talking about homozygous. Then they are making a cross between pure white. Pure white means small r, small r. Now you'll be like, ma'am, how you have decided? We have not seen until now red and white because it is given in the question that red is dominant. That is why I have taken it. Then what will be the ratio of the offspring, right? We'll be making the gamete capital R, small r, then making a cross between them, capital R, small r. That is red. That is 100% of them will be red. The question asked will be all red. I'll be like, ma'am, why they have given 120 plants? Just to confuse you. Just to confuse you, they have given it, okay? Next question, by the way. In a garden pea plant, a pure violet flower is pure violet capital V and a white small v, right? Then they are talking about the F2 generation. Guys, we have already done it, so I'll be writing it very quickly. We have made a cross as well. Remember the diagram which I have taken, right? So the F1 will be capital V, small v, that is all violet. But in the F2, what I'm going to find 1:2:1, that is capital V, Okay. Just give me a second. Yeah. Capital V, capital V, capital V, small v, and small v, small v, right? So, and also I can say 3:1 for violet and white. Guys, I've already drawn it. I've already explained to you. If you want, see, in this type of question, it should be a practice. Just like, since I have drawn this cross multiple times, I know what will be the conclusion. You try to do it. It will be fixed in your own mind, right? I told you the whole concept. Try to do it. It will be fixed in your mind. Even if you practice it twice or thrice, it will be fixed. Just practice, right? So this will be the thing. Let us find out that how many of them are pure if total are 1000? Pure. How many are pure? Pure means, what do you mean by pure? Pure word means homozygous. Now have a look at it. How many are homozygous? How many are homozygous? Homozygous. Homozygous. Out of 4, how many are homozygous? Out of 4, 2 are homozygous. And how many were total? 1000. Just apply your maths, guys. Apply your maths. 2 and we have 5. So obviously the answer will be 500. Right? How many are impure? Impure means heterozygous. Heterozygous. So how many are impure? This one. Capital V, small v. That is out of 4, only 2. Out of 4, only 2. Again, how many are total? 1000. Simple maths. 500. So the answer is 500. Last question. How many are white? White are only 1. Out of 4, only 1. Right? 1 by 4 into 1000. That gives us 250. Right? Answer is 250. Right? Clear? So this was the question. Let's move ahead. The allele which is unable to express itself in the presence of another is recessive. The another should be in the presence of dominant, right? In the presence of dominant, it is unable to express. Which one of these is recessive? Try to do it. You'll be able to find out the answer. Homework from my side. Lastly, okay. Again, violet and white. What will be the result in F1? We all know it will be all violet. 100% violet. Capital V, small v. This is the direct answer. Try to do it. Again, I'm giving you as homework. Try to do it. Next question. Okay, we have a different question here. We have taken BB. We have taken BB. And this BB is nothing but blue. And we have crossed with white. White will be obviously small b. Right? Then color in F1, F2, everything is asked. That is, we have to make the complete monohybrid cross. We will be making it first. We will be making the gametes. What will be the gametes here? Only capital B. The pair will segregate, so only small b. These two will cross pollinate. Cross pollinate. So what will be in the F1? It will be capital B, small b, or we can say all blue, right? So have we got the answer of the first question? State the color of the flower you expect in the F1 generation. All blue, right? The color will be all blue, right? This was the first part. Let's move to the next question, next part of this question. Next question says about the F2, right? So let us draw the F2 also. Capital B, small b crossing with the capital B, small b. The gametes will be capital B and the small b, capital B and the small b. F1 into F1 gametes, right? Then what we do? We make a Punnett square. Let us make a Punnett square here. Capital B, small b. Horizontally in the row, I have kept one type of gamete. Here we're going to write the other ones. Make the combination and put them in the box. So obviously out of these three are blue, and this one is white. So I can say the phenotypic ratio. What will be the phenotypic ratio? How many phenotypes are there? Ma'am, 2 phenotypes are there. That is blue and white. How many are blue? Count it. 3 are blue. How many are white? Only 1 is white. Right? If I talk about the genotypic ratio, what will be the genotypic ratio? How many genotypes are there? 3 types. Capital B, capital B, capital B, small b, and small b, small b. So 1:2:1. Let us look at the question. What must be the percentage of the white flower in the F2 generation? White percentage. What will be the percentage? So obviously 1/4 into 100. That gives us 25%. If it is as blue's percentage, we can say 75, right? Okay, state the expected ratio of the genotype BB and BB, right? Capital B and small b. Capital B and small b. No, they are asking for the heterozygous and the homozygous. So 1:2. This one is 1, this one is 2, 1:2. Easy peasy, isn't it? Capital B is 2. Capital B is 2, capital B, small b. These 2 ratios they have asked. That is 1:2. Right? Chalo, let's move to the next question. Okay, we have done it. The cross between the tall and dwarf. What will be in the F2 homework? You'll be able to do it, right? Lastly, name the 2 types of gametes produced by men. The one having the sperm, one will be having X, one will be having Y. Does a male child inherit X chromosome from father? No. Because the X is inherited from mother. The male child inherits the X chromosome from the mother and it inherits Y chromosome from father. Y chromosome from father, not the X chromosome. Right, guys? Great. How many types of gametes are produced in human female? Only 1 type of egg is there. That is having only X chromosome. Right? That was all about it, guys. So what we discussed today? So today basically we focused on this chapter called as heredity. Obviously not evolution. We have discussed the monohybrid cross, the dihybrid cross, sex determination, and also few questions. The only trick shall I say is practice. Practice. The more you practice, the more you succeed. So your success ratio is dependent on the practice. So please practice more. If you're going to practice well, you're going to score amazing marks definitely. Right? If you have any query, any question coming in your mind, or not able to do any homework which I give, do let me know in the comments. I'll be trying to help you. So that was all from my side to you guys. Thank you so much. Keep learning and stay awesome.